Hello guys, my name is Dr. Varun and I'm back with 30 days, 30 most important topic for NEET 2025 Biology.
Chalo, let's complete it.
So in this today's video, we're gonna be studying about the principle of inheritance and variation one shot. But first of all, I want to make a disclaimer before the start of the video. That is, this video is a quick revision. I'm not gonna be explaining a lot of things. So if you're looking for explanatory videos, then you might go to some other page because this is not that. We had just a very quick glance of everything so that we remember that okay, this thing we have to remember. Next thing, statement based question will come from here. Every year coming, students are struggling, so understanding is very important, beta. If you didn't had an understanding, go to another video first and then come back, take a revision of this, but do that. Starting with the video, that first thing is that the chapter is divided into certain segments, and we're going to see that from initiation to end how much we have understand from the point of view with the principles of inheritance and variations. Starting with the early work, the earliest work regarding the genetics was done by the Mendel, but Mendel didn't get a lot of supports. Let's just quickly see that what we have studied in the Mendel and could be asked to you if the direct questions are there. First of all, the Mendel's law of inheritance. So Mendel is a monk and all those things. They hardly gonna ask you these things. They could ask you that why he choose pea plant. So he did his experiment on what? Did his experiment on what, beta? Did his experiment on the Passum savum. That is what your pea plant. Okay, why? So the reason for why he chooses this, because it is easily cultivable, means easily breedable, easily cultivable, shorter lifespan, so easily you can see the things, bud pollination is there. So these are the basic thing, okay, which made it easier to find the results in the shorter period of time. Took almost 7 years to this experiment, and then he proposed certain laws. He used contrasting character feature. How he come to know the contrasting character? By seeing the things superficially. But how he said that which is dominant and recessive means he said that one feature which is contrasting, like for example, tall and short. One is contrasting, one is, both are contrasting character to each other. One is different, one is different, extremes. But the one he's saying dominant, one he's saying recessive on the basis of the experiment. Okay, so we will learn according from the laws. So first of all, the contrasting characters that you have to remember. So contrasting traits, total 7 different traits were there, out of which these 7 traits, someone will ask like that how many chromosomes they are present on 4 different chromosomes they are present on. Okay, and someone will ask that if they are present on 4 different chromosomes, there might be certain features of them which are present on the same chromosome. Yes, 3 such characters which we are taking over here was present on the same chromosome. But then the people will ask, if you have studied it, then we will learn it later, that why he didn't had linkage problem. The answer I am telling you here, because he didn't actually cross them. If he would have crossed certain of those characters, he might not able to get the results, the one he published at that time. Coming up to the contrasting characters, number 1 is the, since childhood we are like 10th class we are studying, that is stem height. After stem height we have flower color. So stem height, first I'm writing all the characters. Okay, then we are gonna write the contrasting dominant and recessive one. Then flowers, flower position, pod shape, pod color, seed shape, seed shape, and seed color. Now if someone will ask, say, what are contrasting? So first I'm gonna be writing all the dominant one. dominant character and the recessive character. This you have to remember at least. Okay, so dominant character would be what? It will be the tall. Recessive, height wise, it is dwarf. Then dominant is violet, recessive is color wise white, position axial and terminal. So axial would be mostly present. Then pod shape, inflated, means flat like this, but it would be constricted like this. So another is constricted. This is the constrictions. Then pod color, that is, you have to remember that green and yellow is the recessive. But in the seed, this is the change. What is the change? That the seed color yellow is the dominant one, green is the recessive one. Seed shape round and recessive is wrinkled. This you have to remember. Moving on, the first thing is monohybrid called dihybrid cross. You might have done it till now, so I'm not gonna be going in deep into it. But monohybrid cross is remembered as what? One character we are studying, means one gene inheritance we are studying. So monohybrid cross is what? Monohybrid cross is one gene inheritance. Is showing what? Monohybrid cross. So in this cross, when they are taking two contrasting characters, they are getting their gametes, they are mixing up and forming the structure. That first individual that is formed by the contrasting characters, that known as what? Hybrid. This generation is what? F1 generation. It is also called as filial generation 1. Next thing is that how it will look like. So people will think that it will be in between that height. No, it is showing only one character of the parent. What would be the character? Tall. Because we take tall and dwarf. So this plant is tall. So from here the first law came, that law of dominance or law of recessiveness. Okay, so here only I am telling you this thing. Law of dominance. What law of dominance saying? Law of dominance saying that in F1 generation, hybrid, I'm just writing it as easy language. Okay, I'm not writing proper language, just understand it. Hybrid of contrasting character, of one contrasting character, shows only one character, and that observable phenotype means physical appearance of the thing, that observable phenotype is considered as dominant, whereas other would be recessive. So it can also be written it vice versa type. The thing which is not being seen is known as law of recessiveness. True. So law of dominance or law of recessiveness, it's mean same. Okay, this thing is there. Moving on, in monohybrid cross, when the F1 generation, means the hybrid and hybrid will meet, then what it will produce? Since only one gene is taking, 4 different types of outcome is possible. So what are the 4 different outcomes? So 4 options in which the ratio would be very different. Okay, so we will be writing the phenotypic ratio. F2 generation, F1 generation, everyone will be same. Okay, F2 generation phenotypic ratio means how they will look like. So 3 dominant character people will come and 1 recessive character will come. 3:1 would be the phenotype. Then some will ask what would be the genotype or genotypic ratio. Then for example, if I am taking this case, so means this will be 1, this will be 2, this will be 1. Genotypically we can't see, okay, but we can get an answer. Okay, so this all will look what? Phenotypical dominant. If someone will ask like how I will find out that which one is this genotypically, then for that we do what? We do test cross. So here only we will understand what is test cross. So in test cross, an unknown dominant genotype is present, and we will cross it with the recessive genotype. Now there are two options that is possible. First case, second case. If the dominant one is the homozygous case, then in the progeny which will come, okay, when they will make a cross with the recessive type, all of them, means 100% of them will have a dominant feature. Okay, 100% will have a dominant phenotype progeny. But if the heterozygous condition is there, then we will mix it with them. Then what will happen? 50% will come the dominant trait, 50% will come the recessive trait. So 50% dominant phenotype and 50% recessive phenotype. I hope this thing should be clear to you. This is very important. They can ask you question on this basis. So till now what we have studied: basic understanding, dominance, basic feature, one gene inheritance. Okay, every concept about it completed. Okay, this everything we have completed. There possibly things we have done it one shot. This thing explanatory on the ratio, I assume that you know about the ratio things. If not, then most probably I will be taking the, if you guys are not giving your NEET this time, then next time there will be a proper videos will be there on the channel, so you can navigate through it. Okay, so till this we have completed. Look, so fast, it was not anything much to revise. Coming up, the law of dominance also we have corrected. It has certain factors given in your NCERT. Please go through it, don't just leave it. Okay, so law of dominance also we have understood. Here only we will understand one more law, that is law of segregation. Law of segregation means what? When the gametes will form. So it is related to what? First of all, really important is to understand then where it is being done. So when gamete forms, it says that no contamination occur. Means, for example, if there is one child sitting with another child, so both of they will change each other nature. Okay, they do that. This is how the energy is being exchanged. Here it says that even if two different contrasting characters are present, means for example, if it is a hybrid will have what? Capital T, small t. It won't happen that this capital will make this capital or little bit bigger in height, or this also won't lessen up its height. So this will have no effect. So means when they will segregate out, they will segregate these alleles. These alleles are what? The contrasting characters. These alleles would be in their pure form. Okay, so when the gamete forms, no contamination occurs and alleles segregate from each other in their pure form. So remember this fact, remember this way of remembering it. We have completed till here. Now what happened? Then there are certain problems. Okay, later on when the post-Mendel genetics happen, these two laws, law of dominance, law of segregation, it has been shattered. Let's just shatter it up. Okay, so next thing in your NCERT is what? That first question, how law of dominance shatters. So first of all, we will be studying about the codominance. Okay, for that. So in codominance, what happened? That two different alleles are present and we say that one is dominant, one is recessive. But normally we used to say that now one will show effect, it will be dominant, which won't show the effect, it is recessive. But what happened if both are showing it effects? This happens in case of ABO blood grouping. So in ABO blood grouping, what happened? The A allele is dominant. B allele is also dominant. But when the together are present, only one should be dominant. But here what happened? When both are present in the blood group, then the blood group becomes what? AB blood group. So this is what? Co-dominance. Here, law of dominance used to say what? Only one will show it effect. Here, both are showing it effects. Okay, first thing. Now, we are not going deeper into this topic. We are coming back to the major question. Okay, and then we will deep back into other stuff. Coming up to the main thing, how law of segregation shatters. For this, we have to remember the concept of incomplete dominance. Now, incomplete dominance, what happened? If you remember that there is snapdragon, which is antirrhinum species plant, where it happens what? A red is dominant character, white is recessive character. When a red white mix it makes what? Pink. What is this? Not any of the colors are not visible. It is something blended form of it. This is what? Incomplete dominance. Okay, so this is our incomplete dominance. So that is in antirrhinus species, that is snapdragon. One thing is over. Let's just go deeper into it. First of all, we will study from the incomplete dominance only. In the incomplete dominance, if someone will ask like, what is happening? Why this is happening? So there could be possibly three reasons for it. That the gene which is being forming, it is doing the work in three different ways. Either it is working something, okay, which is normal, efficient, close to it. One thing. Second thing, it is not at all working. And third thing, it is doing mutated, something different is being done. So three different options, genes are working, and that's why the different results are coming up. And this becomes really important because this explanation is given over here. And three different options which I have told you and their explanation. So this you have to do it. Very important for you. Here only I am telling you this thing. So incomplete dominance is like that. In co-dominance topic, you will remember that we have done the example of what? ABO blood grouping, right? In ABO blood grouping, it is also the example of multiple allelism. Okay, this is another thing which is not related, but we have to study in terms of, because it's given. That is what? Multiple allelism. Now, multiple allelism will do what? That for a one character, multiple alleles are present in the population. Not in the person. In the person, only two alleles are present. But in population, its other variants are also present. Like you know what? That A, if the A allele, like for example, the person having A, it has either A means both the segments will have AA. The person will be what? A blood group. If it has A and O is what? O is a recessive one. Okay, so it is still the A blood group. Then if it is BB, it becomes B blood group. BO, B blood group. OO, then O blood group. Then we have AB, then AB blood group. So if we look at from the point of view of the genotype, okay, genotype, there are total what? 6 genotype. But if we look forward from the point of phenotype, character, so this is what? A only, this is what? B only, then we have O, then we have AB. So how many of them are present? 4 phenotype. This thing we have to remember. Now this 6 different genotype or 4 different phenotype, because in a person either it will be one phenotype or the other phenotype, like tall or short. But how come here the 4 are there? So in a one person 4 can't be visible. It can only be seen in the population. That you have to remember. Let's just go through one more example which can be asked to you, and I am over here helping you out in making an easy table for it. So that is what with respect to the starch grain. So basically what happened in the pea seed. the starch grain. So one is what seed will have a shape. So in terms of shape, what is there? For example, if capital B is there, capital B small b is there, and small is there. In terms of shape, the capital BB will give what? Round. Small bb will give wrinkled shape, because this is the law of dominance we have already studied. But capital B small b is coming means it is giving what? Law of dominance. So it is what? It will be round in shape. Okay. Then what about the starch grain? Starch grain means it is amount of content that will be present into it. If it is a capital B, then the starch grain size would be of bigger. Okay. So it is a big size. Whereas if wrinkled would be there, small size. Here what should happen? According to law of dominance, it should be big only because capital B is coming. But no, here incomplete dominance occur and it is of intermediate size. So intermediate is what? In between. Whenever in between result is coming means incomplete dominance. Okay. So intermediate size means incomplete dominance is seen here. So we have seen almost all the possible examples over here. Okay, moving on to the next thing, that is 2-gene inheritance. The 2-gene inheritance means what? It is talking about dihybrid cross. Means dihybrid cross. So in dihybrid cross, what would be the phenotypic ratio if you remember? The phenotypic ratio only you have to remember. Genotypic ratio, it's a vague one. Okay, big one. You don't have to. The phenotypic ratio is 9:3:3:1, where this 9 and this are the parental combination. So parental recessive. This is parental dominant. And these are what? These are the recombinants. This thing you have to remember, that how many if they are writing it right, write this phenotype in terms of parental and the recombinants. Then 10:6 would be the answer. Moving on, this thing told us one more thing. That is the next law. The law is what? Law of independent assortment. What does independent assortment means? It means that once the gametes are being formed, the male gamete is free to go and assign with any of the gamete, even of the same type plants. Okay, so this is what it's telling, that gametes, or in total alleles, are free to assort or align themselves to any. Means, for example, if this is the plant species, it will have what? Capital R small r, it will have capital R small r. So now this capital R of this, for example, this is the male plant, this is the female plant. This male plant has an option to go either with this or this. So this is the first option, second option. Same goes with this small. This can go with this can go with this. So it also has two options. So this is the thing. There's no compulsion, anything, any cross can form, and that is what it comes up the probability also in the biology. So that's how probability also ended up into it. Next thing would be that if someone will ask you that how many gametes will be formed from the different types of crosses. For example, in the monohybrid cross, one plant, we have to just remember one plant. Okay, monohybrid cross, they will make two types of gametes. How we will know that? Like, we will know it, like, for example, if you see that this is what? Monohybrid, one gene only I am taking, so two gametes only forming, either capital or small type of genes could be formed. Okay, types. Okay, so this is what? 2 raised to power n. N is what? Number of heterozygous alleles present. Okay, for example, if it is a dihybrid cross, you remember that you used to make 4 different alleles for each parent. So this should be 2 raised to power n about with heterozygous. How many heterozygous things were there? 2, because 2 genes we are taking. So 2 raised to power 2. So that is 4. If someone will ask about trihybrid, in trihybrid 2 raised to power 3, that is 2 into 2 into 2, that is 8. That also okay. Next question they can ask you that how to find the gametes. Okay, so if they are giving you any dihybrid thing, you will be using a Forklin method. Okay, so next is how to find out types or the genotype of gametes formed. Like I said that it will have 2 gametes, it will have 4 gametes, it will have 8 gametes. But how to find it? Let's take an example of trihybrid cross only. So A small capital C small c. So first of all we will divide it all the things. First is what? A. So we have capital A, small a. Now this capital A has option to go with capital B or capital C. We write it like this. So capital B, small b. Then this capital B has option to go with capital C, small c. This also has an option to go with capital C, small c. Same way this small will has an option to go with this. So I will write it capital, small. Then this has an option to go with capital, small. This has an option to go with capital, small. So how many options formed over here? One is this A, capital B, this. Second will be this A, B, small c, A, small b, capital C, A, B, C, all smalls. Then small a, capital, small a, capital, small, capital, small. So these are the different types of gametes that are being formed. All are very different. Sorry, this is A, capital A. So all are what? All are very different. That's it. So this is different types of gametes also. They can ask to you that whether this gamete will form, how many types of gametes, what all the gametes will be formed. So you should know about it. Moving on, till now what we have covered. Till now we have covered this incomplete dominance, codominance, multiple allelism. We have studied the inheritance of 2 genes. This whole thing is being done. Independent assortment has been done. Now this all thing is being told to us by the Mendel, but at that time the time was of the Darwin, so everyone was busy in understanding the Darwin theory, so no one paid attention to the Mendel and his work, which he published in 1865 if I'm not wrong. Yes, it got in the dustbin, not in the dustbin but under the piles of the research papers who no one wants to see. But after a certain period of a time, that is in 1900, what happened? 3 scientists, researchers, they actually find it out about this thing on the basis of chromosomes. So till now, whatever Mendel is saying, he don't even know that the chromosome is there, genes are there, alleles are there. He just got packets, factors or certain things to it. So who was the 3 people who found it out? They were De Veres, they were Correns, and von Tishmak. And they found it out and they took up this all the features which Mendel told and proved that it is similar. And they were the one for the chromosome, and then there was what? Chromosomal theory of inheritance that was given or proposed. Okay, and this theory, which is the chromosomal theory, they were based on the fact that whatever the Mendel has said, it actually what telling us about the genes and the chromosomes and applied to that also. So in your NCERT, 2 different things were given. One is this diagram and another is this diagram. So this diagram is telling us about independent assortment. This diagram is telling about the law of segregation. Okay, so you have to remember this thing. Okay, you have to do this thing. Now the next thing I have told you, quick thing, and meanwhile we move forward with more interesting discoveries. So what is the next discovery? The next discovery here, another person gave, T.H. Morgan. This T.H. Morgan was what? He was considered as a modern genetist. Mendel is called as the father of the genetist. He did what? He found that, okay, till now we have said that law of dominance is not applicable everywhere. Law of segregation is not applicable everywhere. What about law of independent assortment? Even the law of independent assortment has a limitation, and only one way limitation is there, and that is what? That is at the linkage. So linkage is what? When the closely linked genes are present, even if the crossing over will occur, there are very less chances that they will be segregated out. Chances are there, but very less chances are there. And on that basis, Morgan said that even the law of independent assortment is not universal, but we considered it universal. Okay, law of independent assortment do not applicable to what? Your linkage. Next point is that how linkage is from the related to the recombination. Linkage leads to prediction of more parental combination. Recombination makes new variations. Okay, so means the linkage is inversely proportional to what? Recombination. This recombination is what? Crossing over. And that is inversely proportional to distance also. When two genes are closely present, then linkage will be high. So means distance will be more, linkage will be less. Distance would be less, linkage would be more. Remember this thing, and all those factors work according to it. Let's just see that on that basis two different graphs are given in your NCERT. Let's try to understand the basics. Of course, in detail you can read it. This is the first one, this is the second one. In the first one, if you see that this black lines that you are seeing, those black lines are what? These black lines are the genes. In the first case, genes are present very close. In the second case, if you say genes are far off. So tell me, in this one or this one, where more chances of crossing over occur and the new recombination forms? Means there will be recombinants form. Second one, because they are far off. More the distance, less the linkage, more crossing over. Okay, so here it would be less. So the next question is, on what organism T.H. Morgan did this? T.H. Morgan did this experiment on the Drosophila. And this Drosophila, why I chose? Same question for the P also we took. For Drosophila we'll also took smaller lifespan, easily cultivable, easily breedable. Plus you can see that male and female are distinct, characters can also be observable, 2-week lifespan, easily more generation can be formed, genetics inheritance pattern can be understood. 4 chromosomes only present, not much, kuch is there. Okay, that's all about this T.H. Morgan. Moving up to the next concept in the linkage, that is they can ask you question on the basis of gene mapping. I hope you know that. If not, then let me know in the comment box. I will give a separate video for gene mapping type of conceptual questions. Moving on to the next thing, that is next topic which is there, that is polygenic inheritance. Now what is polygenic inheritance? Polygenic inheritance depends upon, it is a type of a quantitative inheritance. Quantitative means more the particular type of gene is present, contrasting character is present, more would be the look like. Means for example what? In case of human skin color, the skin color is determined by the amount of dominant allele present. And there are total 3 such genes are working. So 3 genes for skin color, means A, A, B, B, capital C, capital C. So this thing, if all the three dominants would be present, the person would be pitch black. The word uses negro, black. And if all of them are the recessive traits, then the person is albino. Okay. If these albino person and a black person will mate, then the probable skin type of the child would be what? The genotype would be like this, and the skin type would be what? Intermediate. And in between there will be as much as probabilities would be there. So there would be what the probability? There will be that there are, like, these are what? 6 dominant allele present, 0 dominant allele present, 3 dominant allele present. But there could be the case in between this to this that 5 dominant allele will be present, 4 dominant allele will be present. Whereas over here the things could go in the way of what? That there are only 2 dominant allele will be present, 1 dominant allele will be present. And people would be fairer, very fairer, darker, lighter shade, or fairer, darker. So this thing is there with the polygenic inheritance. I hope you would have understand the polygenic inheritance. Moving to the next thing which is given, that is your pleiotropy. Pleiotropy means what? Pleiotropy means that single gene is affecting multiple functions. Okay, so example of this is what? The disease phenylketonuria. Now we will learn about phenylketonuria more, but here we will understand basic. Inside your body we need an enzyme phenyl. What was the name of the phenyl dehydrolase? What was that? Phenylalanine hydroxylase. Phenylalanine hydroxylase enzyme. This enzyme is needed for the conversion of phenylalanine to the tyrosine. So this is needed. If this enzyme is, like, the gene must be making it, and if the gene that is making it, it is mutated, then what will happen? This phenylalanine is not able to change into the tyrosine, and it will start getting accumulated in what? In your different body parts, like your brain, which will leads to your mental retardation, in the skin, in the, like, below the dermis layer. So different skin is there, head is there, so different organs are being affected. So we call it what? Like single gene problem showing effects on the joints, on this area, on the brain, mental retardation. So this is what? Pleiotropy. Okay, single gene, multiple effects it are showing. Next thing is the sex determination. What is sex determination? That how we determine the sex in different organisms. Now there was a person called Hecking. He found what? X body. He said that he did experiment on a lot of insects and found what? That in certain insects there are X bodies present which are showing only 50% of them are present there, 50% don't, and those which are present that they are different gender, others are different gender. Later on we come to know that X body that he found out, Hecking, is actually what? X chromosome. Okay, and it was present in what? Into the female body but not in the male body. Okay, so male has only single X, but the female has an extra X layer present. So over here how we will remember this fact, that first type of sex determination is where male heterogamety is there. Male heterogamety means male will be different. Like for example in humans. So in humans what happened? The males are XY, females are XX. Same goes with what? It will goes with the like same is in the Drosophila. The Drosophila also XY, XX. There is what? In case of grasshopper or basic insects, what happen in lots of insects? that the male do not have the gamete present, but the female has the same gamete present. So this condition is also there, that they are having one less chromosome present. I said gamete, chromosome. Okay. Second thing is that there is female heterogamety is also present. Female heterogamety is also present, which is seen what? Which is seen in case of in birds. Where in birds what happened? The male has a particular, like a similar kind of a gene, whereas females has what? A different gene, different chromosome, okay, present to it. So this thing we have to remember. After this, the next thing that we have to remember, that is that in case of honeybee, so I assume that you know about the honeybee. In honeybee cases, what happened? There are total 3 different things. They have what? They have males called as drones, which are just like what? They are used for the sperms, nothing else. They are feeded for the sperm. One time they did that flight, reproductive fly, and then the drones are thrown out from the beehive. Okay. Then we have the queen, probably she's a female. Okay. And the last one is the worker bees, which are also female, but they are reproductively inactive. So what happened? Your female lays egg, okay, and the female will produce the eggs, means the female egg, gamete. So female will produce what? Gamete, which is known as egg. It will be what? Haploid, we know that. Now what will happen? These haploid eggs will be produced with the help of meiosis. So female is what? Diploid. Now in this, when drones will give the sperms, so what will happen? Few eggs fertilize. The fertilized egg will be what? Diploid, because sperm will also be haploid. I have always told you that it will be haploid, diploid. These diploid will do what? They will form the female. Out of this, few will be chosen as future queens. So they will be fed on royal jelly. Whereas rest of them would be what? Worker bees. Moving on to the other parts that we have to understand, that is what happened with the other eggs, other unfertilized egg. These unfertilized egg will change into what? Change into drones. So these drones would be what? They would be haploid structures. And if a haploid thing has to make a gamete sperm, it will make by mitosis. That you have to remember. So means if we say that a statement which was given, that is that the queen has the father and the son. Okay, because they can make, look, this queen can have what? This is what? Unfertilized egg will be changing into males. They will be son. This will have what? This will be formed because there will be sperm coming from a future, like from the primitive thing. So this would be what? This is the case of what? They will have a father also. But the drones do not have father or son. They just have grandfather and grandsons. That you have to remember and understood. After this sex determination, the next thing is the mutation. If we are talking about the mutation topic, then it is what? It is telling that certain genes defects occurs and the disorders occurs. Now, how do we tackle this thing? So we can learn about this with the help of pedigree analysis. In pedigree analysis, what happened? That we will put forward certain signs and symbols. I assume that you remember the signs and symbols. Remember the symbols, it can come in your paper. So these signs and symbols, we assign it and we take the history of the patient, that okay, your father has this, your grandfather has this, something, and they try to come up with the particular effect of a gene that could be possibly occur on the family. So this was there. So in your book, 2 pedigree graphs are being made. One is showing what? It is showing autosomal dominant character, that is myotonic dystrophy. Only one disorder which is given in you. Then another example that is given in your book, that a diagram of pedigree, that is autosomal recessive, that is they have written it for the sickle cell anemia, but it will go with all of them. Okay, both of this. Okay, whosoever the disease is, sickle cell anemia, sickle cell anemia. Now here what you have to remember is that in your paper they can ask you to identify which type of disease it is. They won't, like, they can even ask you that they will give you the pedigree, and then first you have to identify that type, whether it is autosomal, dominant, or what it is, and then they have already given a disease out of it. That is a higher level question they can ask, not more than that. What are the other things they could have asked you in the to find it out? That is what, X-linked or sex-linked dominant, or it is X-linked or sex-linked recessive. Now they can give you any pedigree hypothetical and give you these four options that find it out what type of it is. I am giving you an idea. First you will see one thing, whether it is one character you will take, whether it is autosomal or sex-linked. Sex-linked means in all the pedigree there will be only one gender effective. Either all the circles would be all, or maximum circle would be darken up, or the square would be darken up, not both. Okay, if only one gender is included in the disease, then the disease is X-linked. You able to find out one thing, two options already cut it out. Then you will find whether it is dominant or recessive. How you will find? Dominant means it will be seen in every generation. Means if you make this like this is first generation, this is the second generation, and this is the third generation, this one which I am making. So here also dark, here also dark, here also dark. Means what? In all 1, 2, 3 means it is a dominant character, and it is only one sex is being involved into it, that is circle means female, means X-linked dominant character. Just remember like that. I hope you will able to understand this quick thing. If not, then go back and see it again. This question can come to you in your syllabus. Moving on to the next thing, that is the disorders, last topic of the chapter. Okay, so disorders are divided into two different terms. What are they? Number one is the Mendelian disorders, which can be easily seen with the help of what? With the help of your pedigree. Another is the chromosomal disorders. Okay, now your Mendelian disorders, like two we have already named, myotonic dystrophy, sickle cell anemia. Here we're gonna see few more of them. The first one is that what? Your color blindness. In color blindness, this is what? It is X-linked recessive disorders. This thing you have to remember very much. MCQ direct question can come from this, because this is a confusing things. So X-linked recessive disorder, here what happened, the person's cone cells not able to distinguish either red, green, or both could be there, and mostly the males are affected because this is what the color identification gene is present on the X. Now in female what happened, there are two X present. If the female has both the things, then only she will be affected. But if only present on the one X chromosome, the color blindness, but not on the other will act as a dominant, it will go it, because this is a recessive disorder. Okay, so this thing we have to remember. After that, another disorders that we have. Okay, we are not going very deep into it. Of course, read everything, and if you have any doubt, you can ask me on the comment section. But here I'm coming up with the next diseases. We have to quickly complete it. After color blindness, we have hemophilia. Hemophilia also same, exactly same. And this hemophilia could be of different type of clotting factors which are not present in the blood. If that protein is not being formed, clotting protein, means clotting will be hampered. Okay, so it is also X-linked, and this is seen in the Queen Victoria. She was the carrier of the disease and passed it off the lot of males in the family. So X-linked recessive disorder. After hemophilia, next is sickle cell anemia. Sickle cell anemia, we have already studied what it is. It is autosomal recessive disorder. So what happened in this case, like normally your sixth position is of what? It is of glutamate, but it is being changed with what? Valine. Okay, because of the change in the A, GAG to GUG. So this single nucleotide change, that's why it is also called as point mutation which has occurred over here. It makes what? It changed the morphology of the RBCs. So instead of biconcave, they are what? Sickle cell. Means less space means anemia. Patient will have the anemia. Then after sickle cell anemia, we have the next disease, that is phenylketonuria. Phenylketonuria, we have already studied about it. So it is also what? It is also autosomal, can occur in both male and female, and it is a recessive disorder. After that, the next thing is what? Next thing is the thalassemia. Now thalassemia is what? It is also blood related disorder, but here what happened? Hemoglobin is the problem. Either there would be what? Alpha hemoglobin chain will be not properly being made, or the beta globulin chain is not being. So there are two types of thalassemia: alpha thalassemia, beta thalassemia. Alpha thalassemia means alpha chain is not being produced in a good amount, completely not also present, or little bit amount problem would be there. So that person will have alpha thalassemia. Beta thalassemia would be what? It will be because of the beta chains are not, subunits of the hemoglobins are not being properly made. So alpha are more in the blood. What will happen? Anemia and all of these kind of things will be there. On the top of that, who is the responsible for this alpha chain and beta chain not formed? For alpha chain, it is what? It is chromosome number 16. There are two genes, HBA1 and HBA2. They both are working simultaneously. If the one gene can be affected, both the gene can be affected, or either of the gene could be affected. But if it is a case of that is beta thalassemia, then it is the chromosome number 11 where HBB gene is affected. So this alpha and beta thalassemia, it is also what? It is also autosomal recessive disorder. In sickle cell anemia, it is what? If it will come, this gene comes like this, sickle cell, then it will be lethal, death will occur. This I forgot to tell over there. I am coming up over here. With this, your Mendelian disorders are completed. In chromosomal disorder, what happened? Either it could increase the chromosome number or decrease the chromosome number. If there is increment or any change in the chromosome number, we call it aneuploidy, decreasement of the chromosome. If there is increment in the chromosome, we call it polyploidy. Here three disorders are given in your book. Number one is the Down syndrome. For Down syndrome, what happened? Your chromosome number 21, it's one extra copy came in the person. So additional chromosome of 21 is there. It leads to what? Its diagram is given. The baby will be moon kind of a shape, circular in shape, widespread eyes, tongue always rolled out, that flat face bones are there. Okay, broad face, flat broad faces are there, and head is very flat, tongue is rolled out, saliva drooling out, palm crease is there, and then multiple loops are present over here, and congenital heart disorders could be there. So these things are seen in the Down syndrome. Okay, the second thing is the Klinefelter syndrome, in which what happened? Extra copy of the X is being given into the male. So this happened, extra copy, and this leads to what? This leads to the having more female-like characteristics in a male. So what will happen? Breast development will occur. That is called as gynecomastia. There is like not a proper presence of what we say, the reproductive structure in the person. Okay, so these further more things could be there. That could be there. And the last one is the Turner syndrome. In Turner syndrome, what happened? One X chromosome is less. Okay, so this happened what? These females are generally strial, shorter neck. They have what? Low IQ, shorter height, less rudimentary ovaries, not proper developed structures would be present. With this, the one shot of this chapter is over. In completing this one shot, my phone said your memory is full, your battery is gone, every possible thing that could be done multiple times. That's why there was multiple cuts in this. and that really take away the peace of your mind. I tried to give you a quick glance of the chapters. Of course, you have to read NCRT again and again. Maybe I might have missed something. But if you like this video, give it a like, share it with your friends, or just maybe not, because it's the tight time to be more selfish. But do give it a subscribe if you haven't. It's very easy and convenient for you to do a subscribe. And well, that's all for this video. I will take a off.